Hello, we are going to go over very simple probabilistic models in the context of genetics, mainly because this is either a refresher, we may already be familiar with these probabilistic models, or this could be a well-suited brief introduction into this type of models. They're not strictly considered mathematical models. They are models of probability, but they combine well with some of the discussions and some of the topics we are currently discussing and the role of mutation on evolution. Our reference for this part will be Chapter 8 by Dr. Edith Sayer and Dr. Carl Joplin of their book Statistics in a Biological Context, and also Chapter 14 from the textbook on biology OpenStack College. So, one of the most fundamental components um, in the material that makes up chromosomes is the acid that we refer to as DNA. And as we had previously discussed, the typical visualization of DNA comes with a shape of a helix, in which there is a double strand, which is made of two long strands of nucleotides that are connected in a fashion that looks like a ladder, right? So that's precisely what we see in this illustration. And more concisely, there are pairs, connected pairs of bases, and these bases are named as typically denoted with letters C for cytosine, A for adenine, T for thymine, and G for guanine. Okay, so these are the bases representing nucleotides, and in a way, these four letters constitute an alphabet. All right, so it's an alphabet with four characters or four symbols only, and this is an important alphabet because the information pertaining inheritance of traits, the so-called genetic information, is contained in DNA. So in the double-stranded DNA, A adenine pairs with thymine, A pairs with T, and C pairs with G. Okay, and this is also both of those illustrations here come from the reference in chapter 14, which you can see in more detail. Now, the type of thing probabilitists, the type of questions that are relevant in probability, are questions that pertain to counting, and specifically counting repetitions. So if we're interested on the so-called frequency, how many times something is repeated, we are here in the context of accounting or keeping a head count of the repetitions of each of these bases. So how many times is each letter in the four-letter alphabet repeated in a nucleotide sequence? This is a question that is typical in the context of probability. And for this, we're going to refer to a website by the National Center for Biotechnology Information. So here is the website for NCBI. This center is funded by the National Institutes of Health. And here is the URL, which you're more than welcome to explore. There is a lot of resources that you can access from this website. So what we would like to discuss here is an example in chapter 8 of Sayer and Joplin, and it has to do with the frequency or the so-called content on these two particular bases, G and C, on the human papillomavirus 18, and also on a gene that is associated with breast cancer. Here the note is BRCA1. And so this data these frequency counts are obtained from chapter 8. And so these are the numbers, the number of times that this base appears repeated on HPV18. And here in the second column, the third column, we have the percentage of frequency of this letter on HPV18. And we have similar information here for the frequency counts and the percentage. This is percentage of frequency or percentage of repetition. If we're interested on the count, the content of this pair GC, then we're gonna add the percentage of frequency of G and the percentage of that frequency of C. So we can observe that in this column for HPV 18, we have nearly, this is almost, this is 21, this is 19. So for HPV 18, this pairing GC is about 40%, while for BRCA1, the GC content is nearly 60%. That comes out by adding these two numbers. So there's a small note to be made here, and is that databases with genome data usually report on this pairing GC because DNA with higher percentage of G and C is more stable than DNA with larger percentage of A and D. And so this brings us to the following context, in which we would be considering repetitions of the same symbol or the same letter. For example, let's say if we have 4 nucleotides that are equally likely, and the nucleotide in each position is independent of the others, then what is the probability of finding a sequence of this type? Okay, this is a very basic question in probability. Probability it has to do with the chance of having a particular outcome, and in this case we have a sequence with let's count. So we have a sequence with let's see, we have 2, 4, 6, 8, 10 characters, and these characters are the same letter, denoting the same base. And so we would like to explore what is the probability of having this outcome. So one thing to notice here is that there are a couple of things may be subtle. They actually mean more than it seems, and is the fact that we're assuming that these nucleotides were equally likely, and then this thing about independence. So equally likely and independence are very strong assumptions in the context of probability. So mathematically speaking, they're no weak assumptions. But then we're going to say, let's suppose this question refers to the sequence that starts on a particular location without concern of for the nucleotide in the previous location. Or suppose that this that is known that the previous location was occupied by a different letter. Okay, so what we need to calculate is P here denotes probability, and in between parentheses, probability of, the probability of having a sequence that has 11 characters, so a sequence with 11 positions, such that the first 10 positions are the same character, with the letter A denoting the base adenine. And then the 11th position is A to the C here. A to the C denotes a letter that is different from A. Remember, we're working with an alphabet that has 4 characters. And so we are considering a sequence with 11 positions such that the last position is a letter different from the letter that has been repeated, previously repeated. Okay, so in this calculation, P of A denotes the probability of having the letter A, and there are 4 possible letters in this alphabet, and we're assuming this equally likely business, right? So this means unbiased, they all have the same chance. These letters have the same chance of appearing. Then there are 4 of them. Having the letter A is chance 1 out of 4. That means that having a letter different from A would be 1 minus 1/4, which is 3/4. Okay, so we have computed here the probability of having A and the probability of not having A. So the probability of having this sequence with 11 positions would be using the assumption of independence would be the probability of having A times the probability of having A times the probability of having A. So that's 1/4 times 1/4 times 1/4, and that is repeated 10 times. And the last probability is 3/4, right? So that's where this number comes from. We have 1/4 multiplied by itself 10 times. And then that is multiplied by 3/4. And this gives us a number that is has order of magnitude 10 to the minus 7. It's a very large, excuse me, it's a very small, it's a very small number. So what we learn is that longer repeats of the same letter are less likely to happen than shorter repeats. If we computed the probability of a repeated sequence with 4 repetitions of A, is the same type of calculation, but now we multiply 1/4 by itself 4 times, and then that is multiplied by 3/4, and we get a number. This would be 2.9 10 to the minus 3, right? Compare that number 10 to the minus 3, compare that versus a number that is with order of magnitude 10 to the minus 7. So this probability is much larger than that probability.